In an era of increased pathogenic resistance, it is more important than ever to find novel compounds which can combat diseases such as cancer and bacterial infection. You may be able to imagine these compounds being synthesized in the lab, but did you know that many of them were discovered by looking to nature? Meet the Streptomycetes, a diverse genus of filamentous bacteria. Members of this genus produce thousands of secondary metabolites, which are organic compounds including antibiotics, which are not directly involved in an organism's life functions. These compounds are not only useful to the Streptomycetes, but to us as well. In fact, 2/3 of all antibiotics used by humans were originally produced by a member of the Streptomycetes genus. However, issues such as antibiotic resistance are making many of these compounds obsolete. The promise of the Streptomycetes seemed to be coming to an end until the advent of genome sequencing and in silico prediction. These methods allowed scientists to take a closer look at what Streptomycetes really had in store, and what they found was incredible. Most Streptomycetes species have thousands of gene clusters encoding secondary metabolites which scientists had never before seen. In other words, there are thousands of antibiotics, anti-cancer drugs, and more that Streptomycetes have the potential to produce, but just don't. The main question now is, how do we get these compounds to be produced? To understand this, we must first understand why and how antibiotics and other compounds are produced by organisms in the first place. Antibiotics are a specific type of secondary metabolite which evolved as a result of interactions between microorganisms. When two organisms fight for the same resources, the production of antibiotics by one of the organisms can inhibit or kill the other organism, thereby eradicating the competition. In a lab environment, this interspecies competition is virtually non-existent, and there is no need for an organism to produce any of its secondary metabolites. This is where co-culturing comes in. Co-culturing is the act or process of growing two types of living material in a prepared nutrient medium. The result is that inhibitory compounds are produced by either one or both species, which can then be analyzed and commercially produced. Another way to trigger secondary metabolite production relies on the age-old saying, "You are what you eat." The medium in which a microorganism is grown can play an important role in activating signaling pathways which cause the expression of once dormant gene clusters. This approach is based on the fact that many of the chemical compounds found in Streptomycetes' natural habitat is not found in culture media. For example, the compound N-acetylglucosamine, which is normally found in the Streptomycetes habitat, was found to trigger and control antibiotic production when it was added to the culture medium that the Streptomycetes were grown in. Other approaches include adding low concentrations of rare earth metals to the culture media. Harnessing the natural abilities of microorganisms to produce cures to diseases is a promising field in science, with hidden genetic gems just waiting to be discovered. Thanks for watching.